Beringia has, for countless millennia, symbolized humanity's most extraordinary voyage. This land bridge, which once linked Asia to the Americas, was the pathway our forebears traversed into a new world during the Ice Age. While recent decades have introduced theories of coastal migrations, Beringia has consistently retained its central role in explaining how humans initially arrived in the Americas. Nonetheless, despite generations of archaeologists exploring this ancient terrain, unearthing stone implements, animal remains, and remnants of early settlements, the actual human remains of these pioneer travelers have remained incredibly elusive. Today, much of Beringia lies submerged beneath the frigid expanse of the Bering Sea, its secrets engulfed by rising ocean levels at the close of the Ice Age. Yet, a discovery in central Alaska would radically alter our understanding. In the summer of 2013, archaeologists at the Upward Sun River site in Alaska's secluded Tanana River Valley, reachable only by helicopter, unearthed something both profoundly sorrowful and scientifically groundbreaking. Beneath layers of sand and silt at the base of an ancient dwelling, they found what appeared to be a simple burial pit, but its contents would dramatically reshape the narrative of the Americas. Two minuscule skeletons, meticulously laid side by side, having rested in the darkness for 11,500 years. The indigenous community bestowed upon them beautiful names: Sahiti Anen Tidai, or Sunrise Child Girl, and Yukanan Tidai, or Dawn Twilight Child Girl. One infant, later identified by scientists as USR-1, endured a mere 6 to 12 weeks in the unforgiving Alaskan wilderness before succumbing. The other, USR-2, never drew a single breath. A late-term fetus, approximately 30 weeks gestational age, either stillborn or dying shortly after premature birth. Both were enveloped in a thick layer of red ochre, a mineral pigment often employed by ancient peoples globally in burial rites to honor the departed. The anguish of these ancient parents was palpable, transcending millennia. Accompanying the infants into eternity were 4 decorated antler rods, 2 exquisitely crafted lithic dart points, and additional stone tools, weapons and implements that had once felled the colossal game animals sustaining these early communities. Someone had deliberately placed these treasured objects with the babies, perhaps with the hope that they would be needed in another realm. The burial pit was situated directly beneath another hearth feature, one containing an equally tragic finding: the cremated remains of a 3-year-old child, discovered in 2010, dating to the same brief period 11,500 years ago. 3 children, 3 distinct deaths, 3 separate burials, yet all originating from the same moment in time. But therein lay the enigma that would baffle researchers. Why were the infants interred with their grave goods intact, while the 3-year-old was cremated? What calamity had afflicted this ancient family? These 3 children collectively represent the sole confirmed human remains from that era ever found within the vast expanse that was once Beringia. Across that entire frozen landscape, thousands of square miles that once unified 2 continents, only 3 small bodies have endured to recount their story. The true revelation, however, would not emanate from the bones themselves, nor from the exquisite artifacts interred with them, but from something imperceptible to the naked eye: their DNA. A team from the University of Utah, led by anthropology professor Dennis O'Rourke, undertook the arduous task of extracting and analyzing genetic material from the infants' delicate bones. Their focus was on mitochondrial DNA, the genetic material housed within mitochondria, the minuscule powerhouses of living cells that possess their own DNA. This type of DNA is inherited exclusively from mother to child, without recombination, establishing clear maternal lineages traceable through the mists of time, like an unbroken thread connecting past to present. Despite the remarkable age of the remains, older than Stonehenge, older than the pyramids, the team successfully sequenced complete mitochondrial genomes from both infants. From USR1, they obtained 20,004 high-quality DNA sequences. From USR2, an astonishing 32,979 sequences. The cremated remains of the 3-year-old child could not be genetically analyzed. The intense heat of cremation had obliterated any DNA, leaving only unanswered questions. When researchers analyzed the sequences from the 2 infants, they made a discovery that reverberated throughout the scientific community. The 2 babies, interred together, laid side by side with such evident care, belonged to 2 entirely distinct mitochondrial DNA haplogroups, major branches of the human genetic family tree. These children were not sisters. They were not even closely related through their mothers. USR1, who had lived for 6 to 12 weeks, carried the C1b lineage, a genetic signature found today among indigenous peoples from North America's Great Lakes and northeastern woodlands, among the Anishinaabe, Ojibwe, and Abenaki, and dispersed across tribal communities throughout the Amazon basin in Brazil, Peru, and Colombia. USR2, the stillborn, carried the B2 lineage, another distinct Native American genetic marker that appears in indigenous populations stretching from the Arctic regions of Alaska and northern Canada through Mexico and Central America and all the way into South America, down to Patagonia at Chile's frigid southern tip. It's not common to find infants buried together who are not related maternally, O'Rourke remarked, his words laden with implication. It raises questions about the social structure and mortuary practices of these early people. Were these babies cousins? Did they share a father, even though their mothers came from different genetic lines? The burial suggested an intimacy, a connection that transcended mere biology. To grasp the full import of this discovery, we must consider the broader enigma of Native American origins, a mystery that has captivated researchers for generations. Indigenous peoples of the Americas belong to 5 primary mitochondrial DNA haplogroups, denoted by simple letters that belie their complexity: A, B, C, D, and X. The presence of these 5 haplogroups had, for decades, raised an obvious and perplexing question. Did this diversity stem from multiple waves of migration from Asia at different times, with each wave introducing a distinct haplogroup, like ships arriving at various ports? Or could there be an alternative explanation? Some researchers had proposed an alternative hypothesis, which came to be known as the Beringian standstill model. It posited that a single major wave of people journeyed into Beringia from Asia, and resided there not for years or even decades, but for thousands of years. During this prolonged isolation, completely cut off from their Asian ancestors by vast distances, brutal climate, and massive ice sheets 2 miles thick that blocked all southerly passage, the Beringian population underwent genetic differentiation. New mutations emerged and propagated throughout the isolated community. Genetic drift generated variation. The 5 haplogroups did not represent 5 separate migrations from Asia. Rather, they embodied genetic diversity that evolved within Beringia itself, in a population marooned between 2 worlds. The Upward Sun River infants furnished the first direct evidence for this model. Both C1b and B2 are prevalent in Native American populations dispersed across 2 continents. Yet, they are entirely absent in Asia, even in Siberia, where the ancestors of Native Americans originated. This strongly suggested these genetic markers arose after the ancestral population had entered Beringia. Utilizing molecular clock techniques, a method that estimates the age of genetic lineages by measuring the rate at which mutations accumulate over time, like grains of sand in an hourglass. The team estimated that C1b emerged approximately 12,800 years ago, and B2 around 12,000 years ago, after entry into Beringia, but before expansion into the Americas. The infants were living proof that genetic diversity existed in Beringia before the great southward migration commenced. But then came an even more astonishing discovery. When an international team led by geneticist Eske Willerslev at the University of Copenhagen sequenced USR1's complete nuclear genome, all of her DNA, not just the mitochondrial material. The results left researchers stunned. USR1 was genetically distinct from all other Native Americans, both ancient and modern. Her DNA revealed that she belonged to a unique ancient population that scientists had previously been unaware of. They christened this ghost population the ancient Beringians. While USR1 carried this ancient Beringian lineage, USR2 belonged to a lineage ancestral to all other Native Americans. These two babies, interred together with such tenderness, represented two separate branches of the human family tree, branches that had diverged 20,000 years ago, 20,000 years of separation. Yet here they lay side by side. Through sophisticated demographic modeling, researchers constructed a timeline spanning continents and millennia. Approximately 36,000 years ago, a population began to separate from East Asians, though genetic exchanges persisted until roughly 25,000 years ago, indicating that by then they had crossed into Beringia but maintained some contact with their Asian relatives. Between 25,000 and 20,000 years ago, something profound occurred. The Beringian population intermingled with another group known as ancient North Eurasians, people more closely related to modern Europeans than to Asians. This admixture created the ancestral Native American population, the bedrock of all indigenous peoples in the Americas. Then, around 20,000 years ago, the population split. One branch became the ancient Beringians, represented by the little sunrise child girl. They remained in Alaska for thousands of years, adapting to conditions so extreme they defy imagination. Temperatures far below zero, perpetual winter darkness, a landscape dominated by mammoths and colossal bears. The ancient Beringians thrived here for at least 8,500 years, maintaining their unique genetic signature with minimal intermingling with other groups. Meanwhile, the other lineage, from which USR2 and all other Native Americans descended, eventually established themselves further south as the ice sheets receded. But what became of the early Bingians? Genetic evidence indicates that approximately 11,500 years ago, around the time Sunrise Child Girl passed away, individuals with northern Native American ancestry trekked northwards, re-entering Alaska in a reverse migration. This reverse movement might have either displaced or assimilated the ancient Bingian population. Their distinct genetic trace vanished from the historical record, much like smoke dissipating in the wind. Even though elements of their material culture, their tools, methods, and knowledge persisted in later archaeological sites. The ancient Bingians had endured for nearly 10 millennia in one of Earth's most challenging environments, only to disappear. However, even as these genetic findings corroborate the Bingian standstill model, fresh archaeological data complicates our understanding of how humans genuinely entered the Americas. The ice-free corridor, once believed to be the primary route south between the colossal Laurentide and Cordilleran ice sheets, did not become biologically viable and capable of supporting human life until roughly 13,800 years ago. Yet, humans had already reached locations in southern South America by at least 14,600 years ago. The timeline simply doesn't align. The corridor was impassable when people needed to use it. This bolsters an alternative theory, the Kelp Highway hypothesis, which posits that maritime populations navigated down the Pacific coast in boats, sustained by abundant marine resources like fish, seals, and shellfish. Nevertheless, much of this ancient coastline is now submerged hundreds of feet beneath the seawater, making its study nearly impossible. The evidence lies drowned beneath the waves. The simplistic schoolyard notion of Beringia as merely a land bridge that people traversed has been proven far too reductive. Beringia was not merely a thoroughfare connecting two continents. It was a homeland, a sanctuary, a crucible where Native American genetic distinctiveness was forged over millennia of isolation. It was a place where children were born and died, where mourning parents interred them with red ochre and cherished possessions, where entire communities lived, adapted, and ultimately faded away. The infants from Upward Sun River are at the heart of these profound discussions. They represent the most critical human fossils from Beringia, a region immensely vital to human prehistory, yet yielding remarkably few ancient skeletons because most of the land is now submerged beneath the Bering Sea, covered by glaciers, or consists of acidic soil that degrades organic material. Ben Potter, the University of Alaska archaeologist who uncovered the remains, emphasized their unparalleled significance. There are limited ancient human remains in the Americas, and only a tiny fraction has undergone genomic analysis. This represents the first genetic information from the last ice age in Beringia. These infants are virtually the only remains from this crucial period with high-quality genetic data. Without them, we would still be speculating about ancient Beringian populations, the timing of the divergence between different Native American groups, and whether people lingered in Beringia or traversed it swiftly. Without these two tiny girls, who never saw their first birthday, we would know almost nothing about a population that survived for 10,000 years. As research advances, new discoveries and enhanced analytical techniques will refine this narrative. Every additional study, every new ancient DNA sample, and every improved computational model may unveil clearer timelines, more precise migration paths, and a deeper comprehension of population interrelationships. The story of the initial peopling of the Americas will continue to evolve, and more importantly, improve. But the enigma persists. What calamity befell that ancient encampment 11,500 years ago that 3 children perished in such rapid succession? Why was one cremated while 2 were buried? Were they victims of disease, accidents, or the harsh environment? We will likely never discover the full story of that sorrow. What we do know is this: Sunrise Child Girl and Dawn Twilight Child Girl, who lived and died before recorded history began, have bestowed upon us something invaluable. They have offered us a window into our own origins, into the courage and resilience of the people who first arrived in this hemisphere. They remind us that behind every scientific discovery, every genetic haplogroup and demographic model, there were real people who loved their children enough to bury them with the finest possessions they had. Thank you for watching. If you found this video informative, please consider liking it, subscribing, and leaving a comment with your thoughts or questions.